Good afternoon. Right. I want to recap some of the salient points of yesterday's lecture before starting today's. Yesterday we looked at the organization of the levels of organizations in the human. We saw that it starts from the chemical level to the cellular level, tissue level, organ level, organ system, and the human being. So it starts from the basic building blocks to a complex multicellular organism, which is the human being. We then looked at total body water. We said that a percentage of the human being, say 60% of the adult human being's body weight is water. And the total body water declines from infants to it stabilizes in adults. So that the total body water varies with age, gender, and degree of obesity. We saw how the body fluid compartments were measured. And we saw that the total body water was divided into two main fluid compartments, the intracellular fluid compartment and the extracellular fluid compartment. The intracellular fluid compartment forms 2/3 of the total body water, whilst the remaining 1/3 is extracellular. The extracellular is further divided into 3 subcompartments. These are the interstitial fluid compartment, which forms 3/4 of the extracellular fluid compartment. The remaining 1/4 is plasma. And there is this small volume which is allocated to the transcellular fluid compartment. We saw the various fluid compartments, and we discussed how these fluid compartments can be measured using various dyes. We also looked at the characteristics of the various dyes. To qualify as a dye, the dye should have certain characteristics. Then we went ahead to look at the plasma membrane. We saw that it's a lipid bilayer. It's a mosaic of lipid, protein, and carbohydrates. We saw the various lipids that constitute the lipid bilayer. We saw that there are phospholipids, there is cholesterol, and we have glycolipids. Then we saw the proteins. We have peripheral proteins and integral proteins. And we saw the functions of the proteins in the plasma membrane. Next, we looked at the carbohydrates. So that carbohydrates form just a little amount of the plasma layer. And we saw one major characteristic of the plasma membrane is the fact that it is asymmetrical. Now, this afternoon, we are going to start our lecture by looking at membrane transport. That is how substances are ferried across the membrane. Mind you, we said that the fluid compartments, intracellular and extracellular fluid compartments, are separated by a plasma membrane. Now, this plasma membrane is a selective barrier. It allows certain substances to move across it, whilst it restrains other substances. Now, because of this selectivity, the composition of the intracellular fluid is different from the extracellular fluid. Now, permeation of particles across the membrane is dependent on two properties: the relative solubility of the substance in lipid and the size of the particle. Now, if the substance that is supposed to be ferried across the membrane has a high lipid solubility, it is easily moved across. Now, this is because the membrane itself is predominantly a lipid bilayer. It's a lipid bilayer. So lipid-soluble materials readily move across. Now, in addition to the lipid solubility, the other property that affects transport across the membrane is the size of the particle. So as the particle grows bigger, the lower the tendency for it to be moved across. Now, two main transport processes determine the movement of particles across the membrane. These are diffusion and active transport. So the membrane is a selective barrier. Permeation of particles across membrane is dependent on their relative solubility in lipid or lipid solubility, and secondly, by the size of the particle. Doesn't mean you don't have any questions. I can go on. So now we're going to look at the transport mechanisms that take substances across the membrane. First, we look at diffusion. And diffusion is defined as the spontaneous movement of material from a region of high concentration to a region of low concentration. So if substances are moving, there is a net movement from the region of high concentration to the region of low concentration. Then we have diffusion. Now, diffusion across membranes can be simple or facilitated. Now, we're going to look at first simple diffusion. Now, simple diffusion is where substances pass through the membrane without being aided by any integral protein. So here, the substances will be moving from the region of high concentration across the membrane to the region of low concentration. And they are unaided. It means they just go through the lipid bilayer to the region of low concentration. Now, lipid-soluble substances or non-polar substances readily move across the membrane along their concentration gradient. Now, the concentration gradient is the gradient as a result of differences in concentration between the two regions on the either side of the membrane. So substances will move from the high concentrated area to the low concentrated area, and that is described as movements along a concentration gradient or a chemical gradient. The concentration gradient is also the chemical gradient. Now, the cell membrane is impermeable to large water-soluble substances. So large water-soluble substances would have to find other means to get across the cell membrane other than the simple diffusion. Now, ions would use channels to move across the membrane. Remember, we said that in the lipid bilayer are transmembrane proteins. Now, some of these transmembrane proteins are channels. It means that they have balls, water-soluble balls within them that allow ionic diffusion, that allows ions to move from the region of high concentration to the region of low concentration. And that is described as ionic diffusion. Now, this happens because ions do not go through the lipid membrane unaided. They have to use these channels. Now, in addition to the concentration gradient, ionic movement is also dependent on the electrical gradient. Now, I'll use this diagram to explain the chemical gradient and electrical gradient. In A, at the beginning of the experiment, that is before the equilibrium, the concentration of the particles in C1 is greater than particles in C2. And these are separated by a membrane. Now, the movement of particles from C1 is greater than movement of particles from C2. Now, that is movement from C2, that is movement from C1. Now, because of the differences in concentration, there is a net movement of particles from C1 to C2. And at equilibrium, where there would be no net movement, even though there's movement, no net movement. It means movement from C2 to C1 is balanced by movement from C1 to C2. Now, this is the chemical gradient or the concentration gradient. If we come to B, we have the electrical gradient. Now, this part of the membrane is negatively charged. This part of the membrane is positively charged. So we realize that positively charged ions are attracted to the negatively charged region. Net movement. And the negatively charged particles are attracted to the positively charged region till equilibrium is established. Now, ions will utilize both concentration gradient and electrical gradient during movement. So ionic movement is dependent on electrochemical gradient. Unlike these fat-soluble substances that move just along the concentration or the chemical gradient. The ionic movement movement is determined by the electrochemical gradient, meaning the charge of the environment and the concentration gradient determine ionic movement. So that is simple diffusion for you. It is movement across the membrane unaided, right, from a region of high concentration to a region of low concentration. Ionic movement depends on chemical gradient as well as electrical gradients. Now, other substances will move across the membrane from a higher concentration to a lower concentration by facilitated diffusion. It means this is quite simple from simple diffusion. Now, this movement is aided by a transmembrane protein. Now, this is not a channel. This is not a channel. This movement utilizes carriers. They use carriers which are transmembrane proteins, unlike the channels that are utilized by the ions. Now, the channels are transmembrane proteins. The particles to be carried across will bind to the channels. A channel will undergo a conformational change that will ferry the particle across the membrane to the region of lower concentration. So in facilitated diffusion, we still have substances being moved from a region of high concentration to a region of low concentration, but with the aid of a carrier. I would use this analogy. Let's say if you try to move across a river, you'd sit in a ferry or a boat, and this ferry or boat will carry you across the river from one bank to the other. Now, the ferry or the boat will be the carrier. So, you are being aided across the membrane. Now, if you just swam across the river, then you are moving unaided. So, simple diffusion could be like swimming across the river. And facilitated diffusion meaning crossing the river with the aid of a boat or a ferry. Facilitated diffusion does not require energy, right? It does not require energy. And it exhibits some characteristics of carrier-mediated transport. Now, the carrier-mediated transport have some characteristics, and these characteristics are: it is more rapid than the simple diffusion. And I'll go back to my example. So, I'll go back to my example. Swimming across the river and moving across the river in a boat. Moving across the river in a boat is faster than moving across the river swimming, right? So the facilitated diffusion is more rapid than simple diffusion. Secondly, facilitated diffusion exhibits what we call saturation. It exhibits saturation. If we look at this graph, this is simple diffusion. As the concentration of the solute increases, the rate of transport increases in simple diffusion. In facilitated diffusion, as concentration of solute increases, transport rate increases rapidly till it levels off. So at this point, it's exhibiting what we call saturation. Now I'll use an example. Assuming you are traveling from Lagos to Abuja, and you are going on a bus that takes off regularly. You go to the bus station, there is about a 30-seater bus that would have to take off at hourly intervals. I mean, the buses take off at hourly intervals. Now, if the bus is, say, 9 o'clock, and there are only 10 passengers, the bus will have to take off. Now, if at 9 o'clock there are 20 passengers, they all go on board and the bus takes off. At 9 o'clock, if there are 30 passengers at the station, all of them will go aboard and the bus takes off. At 9 o'clock, if there are 31 passengers, 30 will go on board, the one will stay and wait for the next bus. So at this point, the bus is saturated and it takes off. If there are 32, 30 will go aboard, the remaining 2 will stay and wait for the next bus. So in the facilitated diffusion, there are places on the carrier that would move the substances across the membrane. If the places are saturated, no more particle will be moved across. So the carrier is exhibiting what we call saturation. Now, still on the characteristics, it exhibits specificity. Specificity meaning that a carrier will ferry only particles of a particular kind. Right. So let's say if the carrier is ferrying glucose, it will only pick up glucose. It won't pick up any other sugar, be it sucrose or galactose. The carrier will take only glucose. So it is specific. So we say that the carrier exhibits specificity. Now, the carrier also exhibits competition. Now, if there is a particle which is similarly structured to the particle that the carrier transports across, they'll compete for the position on the carrier. Assuming we have D-glucose and L-glucose. Now, the carrier would be moving D-glucose if it were present alone, or will be moving L-glucose if it's present alone. Now, if we have both D and L-glucose present, they will compete for the point of attachment on the carrier to be ferried across. So we say that they are competing on the for the carrier. Now, I'll use an example. Let's say a boys' school and a girls' school. Now, boys will apply to attend the boys' school. Girls will apply to attend the girls' school. Now, for a mixed school, both boys and girls will apply to attend that school. So there is a competition between the boys and the girls. Right. It's the same way similarly structured particles will compete for the same carrier. So D-glucose, L-glucose will compete for the same carrier. And the carrier is set to exhibit competition. Then finally, the carrier-mediated transport also exhibits inhibition. There are times that the presence of a chemical or a particle, which is very different from the particle that is carried by the carrier, would inhibit the activity of the carrier. So we have, let's say, the carrier is supposed to move particle A, and another particle which is not similarly structured as A is present. Once it binds to the carrier, it inhibits it. This particle will not compete with the carrier for its binding parts. It just binds to the carrier anyhow and inhibits the carrier. So the presence of that particle alone can inhibit the activity of the carrier. So this is facilitated diffusion. Now, good. This chart summarizes simple diffusion. Up here, we have these particles that would move directly across the lipid bilayer. Now, these particles, the yellowish, have high lipid solubility. So they'll just move across the lipid bilayer since it is also lipid. They'll just dissolve and move across. Examples of such molecules are oxygen and carbon dioxide. They readily move across the membrane using their ability to dissolve in the lipid bilayer. Then we have the ions, the red particles. Now, these ions are unable to move across the lipid bilayer using the layer. So they utilize transmembrane proteins called channels. So we have a channel, and in the channel we have a ball, water-soluble region where the ions will move across. So the ions will move across utilizing channels. Then we have facilitated diffusion. Now, these two are simple diffusion, right? Then we have facilitated diffusion, where the particles will be moving across using a carrier. Now, this is a carrier. The particle will bind to the carrier. The carrier undergoes a conformational change, right? And the particle is released to the region of low concentration. Then the carrier will assume its original conformation. The particle binds to it again, it undergoes a conformational change, and the particle is ferried across the membrane. Now, in all these cases, the particles are being moved from a region of high concentration to a region of low concentration. So it's moving along a chemical gradient. It's moving downhill. Have I made sense so far? Esther, feedback, please. Esther, awaiting your feedback. Good. So now that deals with diffusion. The other transport mechanism that would move substances across the plasma membrane is active transport. In active transport, the substances are moved from a region of low concentration to a region of high concentration. In diffusion, substances are moved from a region of high concentration to a region of low concentration. In active transport, substances are moved from a region of low concentration to a region of high concentration. Now, this requires energy since substances are being moved from a low concentrated region to a high concentrated region. Now, the movement will require the use of a carrier protein, just like in facilitated diffusion. Now, in facilitated diffusion, it is from a high concentrated region to a low concentrated region. But in active transport, from a low concentrated region to a high concentrated region using a carrier protein, which is a transmembrane protein. So active transport also exhibits the characteristics of the carrier-mediated transport, since both active transport and facilitated transport are carrier-mediated. But one, which is facilitated diffusion, does not require energy because substances are moved from a region of higher concentration region to a low concentrated region. In active transport, movement is from a low concentrated region to a high concentrated region. So that is moving against a chemical gradient. In diffusion, we are moving along a chemical gradient. In active transport, we are moving against a chemical gradient. Now, there are two forms of active transport. These are primary active transport and secondary active transport. I'll explain each one of these transport mechanisms, active transport mechanisms. At least you should remember this. In diffusion, substances are moved along a concentration gradient or a chemical gradient. In active transport, in diffusion, substances are moved along a concentration gradient or a chemical gradient. In active transport, substances are moved against a chemical gradient. So moving from a region of low concentration to a region of high concentration. Unlike in diffusion, where substances are moving from a region of high concentration to a region of low concentration. So now we are the primary active transport. Primary active transport utilizes metabolic energy in the form of ATP, right? Adenosine triphosphate. ATP is adenosine triphosphate. It's the energy source mainly in cells. And examples of active transport processes are the sodium-potassium ATPase, the calcium ATPase, and the hydrogen-potassium ATPase. These are enzymes, transcellular proteins, and they are enzymes. We saw that one of the functions of the proteins in the lipid bilayer or the plasma layer is enzymes. They are enzymes, right? So now I'm going to describe the sodium-potassium ATPase and how it works to you. Now, the sodium-potassium ATPase, which is commonly called the sodium-potassium pump, can exist in two conformations. In one conformation, it has a greater affinity for sodium ions. And in the other conformation, it has a greater affinity for potassium ions. Now, this is a very common transporter in animals. From our body fluid lecture, we know that the concentration of sodium in the extracellular fluid is higher than sodium in the intracellular fluid or cytoplasm. Concentration of potassium in the intracellular fluid is higher than outside or in the extracellular fluid. Now, ions can move across the membrane using channels. So sodium can move along its concentration gradient into the cell using sodium channels. In the same vein, potassium can move from the cell to the extracellular fluid using potassium channels along their concentration gradients. Now, for survival and stability of the cell, the sodium that gets into the cell would have to be pumped out. The potassium that goes outside the cell needs to be pumped in. Now, because sodium concentration in the cell is lower than outside the cell, moving sodium from within the cell to the extracellular fluid will be against its concentration gradient, would be against its chemical gradient, because you are moving from sodium from a region of low sodium concentration to a region of high sodium concentration. In the same vein, if you are moving potassium in, it is moving from a region of low concentration area, that is in the extracellular fluid, to the cytoplasm where it is in high concentration. So that would also require energy. So now let's see how the sodium-potassium pump works. In this conformation, which is the original conformation, the sodium-potassium pump has a greater affinity for sodium ions. So 3 sodium ions from the cytoplasm will bind to the pump. So we'll have this. When sodium binds to the pump, it will utilize ATP. It will hydrolyze ATP. That is, it will break down ATP into ADP and inorganic phosphate. Now, this hydrolysis process energizes the sodium-potassium pump. And when the phosphate binds to it, it decreases the affinity of the pump for sodium ions. So sodium will be released from the pump to the exterior of the cell. All 3 sodium particles will be released to the exterior. Now, this conformation favors the binding of potassium ions. So potassium will then bind to the pump. When potassium binds to the pump, the inorganic phosphate that is originally attached is released. And when the phosphate is released, the pump assumes its original conformation. Without the phosphate, the affinity for potassium declines, and the potassium is released into the cell. Now, when potassium is released into the cell, 3 sodium will then bind to it as long as there is ATP. So the conformational changes ensure that sodium is moved out of the cell against its concentration gradient, and potassium is brought into the cell against its concentration gradient. Now, this is a bit confusing, so I'll go over it again. Please just pay attention. In the original form, the particle has a greater affinity for sodium ions. So it's sodium loving. Sodium then binds to the particle. And this undergoes a conformational change. In this conformation, the particle, which is a transmembrane protein, loves sodium. So 3 sodium ions will bind to it. Then we have ATP binding to it, and the ATP is hydrolyzed. This is a high energy particle, ATP. When it's hydrolyzed, it's converted into ADP, an inorganic phosphate. And this inorganic phosphate would energize the particle. Now, so the energy that is gotten from this process will be used to move sodium out of the cell. Right. Now, because of the presence of this inorganic phosphate, the particle undergoes a conformational change and the sodium is released because this particle conformation, does not love sodium, literally. The affinity of this conformation for sodium decreases, so the sodium is released. Now, this conformation is rather potassium loving. So potassium will then bind to the particle in this conformation. Now, when potassium binds to the particle, it undergoes a second conformational change and the inorganic phosphate is released. Once the inorganic phosphate has been released, it assumes the original conformation, which is not potassium loving. So the potassium will be released into the cell. Now, this original conformation is sodium loving, so sodium binds to it and the cycle goes on. As long as there is energy, the movement will continue and sodium will be moved from the interior of the cell to the exterior. So from a region of low concentration to a region of high concentration. While potassium will be moved from the exterior of the cell to the interior. That is from a region of low concentration to a region of high concentration. So because sodium is moved against the concentration gradient and potassium is also moved against the concentration gradient, energy is required, and this energy is provided by the ATP. And because this movement is against concentration gradient, it is said to be an active transport process. And a primary active transport process because it utilizes directly metabolic energy in the form of ATP. Good. So that is our primary active transport. Now let's look at our secondary active transport. In secondary active transport, there's indirect utilization of metabolic energy. Here, energy is not in the form of ATP. Rather, the energy that is used for the movement of a particle against its concentration gradient is derived from the energy that is released by another particle which is being moved along its concentration gradient. Now, example of secondary active transport is the co-transport of sodium and glucose, or the co-transport of sodium and amino acids. Now, let's look at this diagram. This process is a secondary active transport process. Sodium and glucose are being moved from the extracellular fluid compartment into the cell. Now, we know that in the extracellular fluid, sodium concentration is much higher than in the cell, that is in the cytoplasm. Now, the concentration of glucose outside the cell is lower than the concentration of glucose in the cell. So this transport mechanism will move sodium down its concentration gradient and will move glucose against its concentration gradient. Because sodium is being moved from a region of high concentration to a region of low concentration, it releases energy. It releases energy. And the energy that the sodium transport is releasing will be harnessed by the carrier protein to move glucose from a region of low concentration into the cell where glucose concentration is high. So here, sodium will bind to the carrier. Glucose will also bind to the carrier. Now the carrier will undergo a conformational change here. Now during this process, the sodium transport would release energy. Because sodium is being transported along its concentration gradient, it releases energy. And the energy that the sodium transport releases is used by this carrier to move the glucose against its concentration gradient. So that the sodium is moving along its concentration gradient, and so that the glucose is moving against its concentration gradient, utilizing the energy that sodium transport is releasing. So that those of you who have ridden bicycle, if you are riding downhill, you don't need to pedal. So that is diffusion. If you are riding downhill from the top of the hill to the bottom of the hill, you don't need to pedal your bicycle, but you get to your destination. So that describes what we call diffusion. Movement from a high concentrated area to a low concentrated area. You don't require energy. Now in active transport, it's like riding uphill from the bottom of the hill to the top of the hill. And that requires much effort, much energy. You need to pedal. If you don't pedal, there is no way you can move the bicycle from the bottom of the hill to the top. So that requires energy. Right? If you are pedaling, it means you are using energy, moving against the concentration gradient. And we said in active transport, we have the primary, which utilizes direct energy. Direct energy. In secondary, energy is obtained from the transport of a particle from a high concentrated region to a low concentrated region. And the energy that is released will be harnessed by the carrier to move the other substance from the region of low concentration to the region of high concentration. Now, these carrier systems can be unipots or can be sympots. Sympots or antipots. Now, sympots would carry two particles in the same direction. An example of a sympot is the sodium glucose carrier. Now, this carrier is moving both sodium and glucose in the same direction. That is from the extracellular fluid into the cytoplasm. So it is a sympot. Now, the sodium-potassium pump is described as an antipod. Antipod because it is moving sodium in outside the cell and bringing potassium inside the cell, meaning the carrier is moving the particles in opposite directions. So it is said to be an antipod. When it's moving both particles in the same direction, it is said to be a sympod. Now, we can have a unipod. A unipod will move a particle, only a single particle. It doesn't move more than a single particle. So it's said to be a unipod. Now, we can have a co-transporter. A co-transporter will be moving more than one particle. And a co-transporter can be a sympod or an antipod. A sympod is where the transporter is moving the two particles in the same direction, like we saw with the sodium glucose transporter. And an antipod will be moving the particles in opposite directions, like we saw with the sodium potassium ATPs. It moves out potassium and brings in it moves out sodium and brings in potassium. So it's an antipod. Good. So that is our active transport. The next transport mechanism we're going to see is the vesicular transport. Vesicular transport. Now, this is the movement of macromolecules, that is, large molecules, across the membrane. And these molecules are packaged in vesicles. They are packaged in vesicles and sent across the membrane. Now, there are two types of vesicular transport. We have endocytosis and exocytosis. Now, endocytosis is the uptake of particles by the cell, meaning the particles which are to the exterior of the cell will be taken up by the cell. So, endocytosis. And exocytosis is the secretion or release of macromolecules from the cell. It is giving it out, so it is exocytosis. Now, looking at endocytosis. Now, there are three types of endocytosis. That is uptake of macromolecules. We have phagocytosis, which is the uptake of particulate particles. Big particles. The uptake of big particles. Then we have pinocytosis, which is the uptake of soluble substances in the very little particles almost dissolved in solution. Right. So the phagocytosis could be maybe, let's say, taking a bolus while the pinocytosis is taking fluid. Right. Then we have the receptor-mediated endocytosis. Receptor-mediated endocytosis. Now, during endocytosis, the plasma membrane surrounds the particle that is to be taken up and forms a circle around it. And when it's gone around the particle, the vesicle pinches off and it's internalized. So I'm sure you might have done amoeba in your basic sciences. Amoeba is a unicellular organism. As it moves, when it encounters a particle, it sends its membrane around it. and this goes round it to form a vesicle, and the vesicle pinches off and it is internalized. So the vesicle will contain whatever that the organism wants. Now, I mentioned that there are three types. We have phagocytosis, which is the uptake of very big particles, like food particles. An example of that is the uptake of bacterial cells by macrophages. Macrophages are white blood cells. So if the body is attacked by bacteria, these would send their particles around the bacteria, internalize it, and destroy it. So that's an example of phagocytosis. Then we have pinocytosis, where very soluble in solution form. The membrane goes around it, forms a vesicle, and pinches off, and it is internalized. Then we have what we call the receptor-mediated endocytosis. Now, this particular method occurs in special regions in the membrane called coated pits. Now, this is a coated pit. Now, pinocytosis, phagocytosis do not take place in coated pits. Now, the coated pits are regions in the membrane that have bristles. Now, these bristles, these blackish things there, hair-like structures, are formed from proteins called clathrin. The name of the protein is clathrin. So the specialized regions with these bristles are called the coated pits. And in the coated pits are receptors to which the particle to be taken will bind. When they bind to the receptors, the vesicle is formed and this will pinch off into the cell to be internalized. Now, this pinching off of the vesicles depends on the presence of a protein called dynamin. That's dynamin, the spelling. Dynamin here. So if dynamin is present, the area will pinch off. And the process of endocytosis requires energy. Now, exocytosis, which is the other vesicular transport mechanism. And this is how cells secrete their contents. For instance, neurons will secrete their neurotransmitters by exocytosis. Cells will secrete their contents by exocytosis. Release of hormones by glands is by exocytosis. And this requires energy. The process of exocytosis requires energy, and it requires calcium ions. Influx of calcium ions will trigger the process of exocytosis. Now, this chart portrays exocytosis. Would have the content, let's say the hormone, the transmitter or enzyme that needs to be released or secreted, will be packaged in vesicles. Now, these vesicles have plasma membranes. Then the vesicles would move to the plasma membrane, that is the membrane surrounding the cell, fuse with the plasma membrane, and a neck will be opened. And once the neck is opened, the content of the cell will be left outside the cell. So the vesicle is the packaging is done in the cell, and it fuses with the membrane, and there's some form of rupture creating a neck, and the content is released outside the cell, and the membrane remains intact. The lipid bilayer remains intact. Now, this requires energy, and it requires calcium ions. The calcium ions will trigger the exocytotic cytosis process. Now we move to the next transport process, and that is osmosis. Osmosis has been defined as the movement of water molecules across a semi-permeable membrane from a region of low solute concentration to a region of high solute concentration, or the movement of molecules of water from a region of high water concentration to a region of low water concentration through a semi-permeable membrane. Now, this semi-permeable membrane allows only water to move across, but not the solutes, right? Now, the presence of a solute in water would decrease the chemical potential of water, right? Would decrease the chemical potential of water. And when chemical potential of water is decreased, water would have to move into that particular compartment. Now, let's look at this setup. Now, the limbs of this setup is separated by a semi-permeable membrane. The solution here is a weak solution, and that is a very concentrated solution. It means that it has more solute than this portion. This portion has more solute than this portion. Now, the solute in the water decreases the chemical potential of water. So water will tend to move from the region where its concentration is high to the region where its concentration is low. Now, the water concentration in this limb is low because this is a very concentrated solution compared to this one. So the water will be moving in this direction. The net movement of water will be in this direction. Now, water movement continues till there is a balance in concentration, or an osmotic pressure is generated which stops water movement. As water moves from the weaker solution to the concentrated solution, it creates a hydrostatic pressure. Now, when the hydrostatic pressure reaches a certain point so that there is no movement of water, we say that an osmotic pressure has been attained. The osmotic pressure that is attained will prevent further movement from this solution into that. Now, the hydrostatic pressure is also referred to as the water stopping pressure. And the pressure at which the water flow, or there is no net flow of water, is what we call the osmotic pressure. Now, I'll use the second setup to explain this further. If we have this solution, that's water. Water will be moving from the less concentrated solution or pure water into the solution. Some water will be moving into water, but this is very small compared to water moving from water. So we said that there is a net movement of the water to the solution. As a result, the solution here becomes diluted. There is a generation of a hydrostatic pressure. Now, the point where there is no net movement of water is where the osmotic pressure in the solution is attained. That is, the hydrostatic pressure which stops net movement of water is the osmotic pressure. So the osmotic pressure is this height, right? This is the osmotic pressure. Now, if we have our water and our solution, and we apply an external pressure, the external pressure will prevent water movement. And the pressure where there is no net movement of water is the osmotic pressure of the solution. The osmotic pressure of a solution depends on the number of solid particles present in the solution. Now, if we have two solutions, A and B, and they have the same pressure, osmotic pressure, the solutions are said to be isotonic to each other. Now, if the concentration of solute in A is greater than that of B, A is said to be hypertonic to B. Now, if the solution A has a lower concentration of solute compared to B, A is said to be hypotonic to B. So two solutions could be isotonic when the osmotic pressures are equal. Now, one can be hypertonic to the other if the concentration of the one, say A, is greater than B. Now, because the concentration of B is lower than A, B is said to be hypotonic to B. Now, I want to give you a practical way of osmosis occurring in the body. Now, during the giving of infusions, normally the infusion is 0.09%, right? Now, this is isotonic to plasma, right? So that if you are giving the infusion, it's not going to destroy the red blood cells because it's isotonic. Now, if you're giving an infusion with higher than 0.09% sodium chloride, then that will be hypertonic and it can destroy red blood cells. It causes rupturing of red blood cells. Now, it is important that whatever you're giving, it's hypotonic to plasma. Now we move on to the final transport process that occurs, and this is filtration. And filtration, fluids are forced across membranes because of the pressure differences on the two sides of the membrane. So fluids will be forced from the region of high pressure to the region of low pressure. And the rate of filtration depends on the pressure difference. If the pressure difference is big, rate of filtration is high. If the pressure difference is small, filtration rate is low. The rate of filtration is also dependent on the surface area of the membrane. If the surface area is big, filtration rate is high. Then filtration also depends on the permeability of the membrane. So depending on the coarseness of the filter, if the spaces between the filter is big, then filtration rate is high. If it is fine, very low, then it is low. It's like trying to filter something with a sieve. If the sieve is smooth with very little balls, it's difficult to filter. But when it's wide, then it's easier to filter. And an important filtration process that occurs in the body is in the glomerulus. I guess you know where the glomerulus is. In the kidney, we have the nephron, right? And the glomerulus is a ball of capillaries where filtration takes place. Now, the vessels leading to the glomerulus are bigger, and those that would move exiting the glomerulus are small. So there's a pressure difference there, and this forces fluid in the capillaries to get into the Bowman's capsule. I guess when you're doing the renal system, this will be elaborated on, right? And you understand it better. Now, this ends our transport processes. This ends our transport processes. Now, we have 30 minutes and we are going to look at homeostasis. Now, homeostasis is the maintenance of a relatively constant internal environment. The maintenance of a relatively constant environment. Now, the concept of homeostasis was developed by a French scientist called Claude Bernard. He performed a series of experiments and he moved an organism from one environment to the other. And he realized that irrespective of the environment, certain parameters in the organism were kept constant. They did not change. So he proposed this concept of the constancy of the milieu intérieur. That's French, and I'm not good at French, so we carry on. That was in 1878. Now, the concept was elaborated by an American scientist called Walter Cannon. And it was Walter Cannon who coined the word homeostasis from two Greek words. Homeo, which means same, like, or resembling, and stasis, which means to stand or posture. So literally, this means standing same, right? So maintaining the same posture. Now, the internal environment of an organism is the extracellular fluid. Mind you, we have intracellular and extracellular fluid. The extracellular fluid is the internal environment of the organism, and in our case, the human being. The extracellular fluid, meaning the plasma and the interstitial fluid, as well as the transcellular fluid. Now, the essential variables in the internal environment that need to be maintained constant or within limits are the concentration of oxygen and carbon dioxide. So the gases that we breathe in or out need to be regulated. The pH of the internal environment, that is the acidity, needs to be regulated. It shouldn't be off gear. Blood acidity, blood gas contents. The concentration of nutrients and waste products. concentration of salt and other electrolytes, the volume and pressure of the extracellular fluid, and temperature. These are all variables in the internal environment that need to be regulated within limits. It has to be constant. If it varies, then illness sets in. Now, homeostasis is continually disturbed by stress. And stress has been defined as any factor or any process that upsets homeostasis. Any factor, process that would upset homeostasis, that is, throw the homeostatic balance out or cause imbalance, is said to be stress. Now, the stress can be from within or from without. And I guess you can name a lot of stresses. I'm sure some of you are stressed out because of this afternoon lecture, right? But this wouldn't harm you anyway. So let's continue. Now, diseases result from the disturbance of homeostasis. So normal human beings, when we are not troubled, would have homeostasis. But stresses can cause imbalance in homeostasis. Now, homeostasis depends on interaction between the 11 organ systems that we saw yesterday during our lecture on the levels of organization in the human body. The 11 systems in the body, organ systems in the body, are responsible for maintaining homeostasis in the human being. Now, the nervous system, and to a lesser extent, the endocrine system, are the main control systems. So the nervous system and the endocrine system control the other systems to ensure that there is homeostasis. Now, because of our ability to maintain homeostasis, human beings and mammals, for that matter, can live in different environments. Now, if you are taken from your present environment and taken to maybe Greenland, you still survive there because of your body's ability to maintain homeostasis. If you are taken from there to a desert place, you still survive because of the ability to maintain homeostasis. Now, we're going to look at the homeostatic control systems. And the control systems are the components of the body, so the organ systems that we saw. Now, these would ensure or generate compensatory responses to maintain a relatively stable environment. Meaning, if the internal environment is threatened by a stressor, these systems, which are the control systems, would generate a compensatory response to combat the stressor. Now, the control system has three components. They have the detectors or sensors, the integrating center, and the effectors. Every control system has these components: detectors or sensors or receptors. There's the integrating center, and we have the effectors. Now, the detectors, which are the receptors, will detect changing internal environment, the stimulus. So they detect the stimulus and will generate an output. The stimulus will be sent to, the changes will be sent to the control center or the integrating center. Now the integrating center is the brain and the spinal cord. So the receptors will detect the environmental change. The information will be sent via sensory neurons to the integrating center, which is the brain or the spinal cord. Now, stored in the integrating center are some set points for the controlled variables. The integrating center will then compare the information it's receiving from the sensors to its set points. Now, this will bring about any deviation from the set point. So the integrating center will generate an output. The output will be sent to the effector, which will bring about a response to correct the change. I'll go over that again. The change is detected by the receptors, the sensors, or detectors. Now the change is then relayed to the integrating or control center in the central nervous system, so the brain and the spinal cord. Now the information that the integrating center receives is compared to the set point of the various variables. If there is a deviation, an output is generated and sent to the effector. Then the effector will generate the appropriate responses to correct the change. Now, I'll use this to explain this process further. Now, this is the body temperature. The normal body temperature is 37 degrees C, let's say plus or minus 0.5. Right. But it's normally 37 degrees C. Now, if the body temperature should go up, if we go into a hot place and the body temperature goes up, this body temperature is detected by thermoreceptors. Now, these thermoreceptors are temperature receptors. When they detect the change in body temperature, the information is relayed via sensory neurons to the integrating center. In this case, the hypothalamus of the brain, where we have the temperature regulating center. Now, the information that the hypothalamus gathers or receives is compared to the set point in the hypothalamus. And we know that the set point is 37 degrees C. So let's say it's receiving information that the temperature is 39 degrees or 40 degrees. So there's a deviation of about 2 or 3 degrees C. Now the regulatory center or integrating center will generate an output which will be relayed via motor neurons to the effectors. And the effectors will generate the appropriate responses. And what are the responses? First, the blood vessels supplying the skin will dilate, meaning blood flow to the skin will increase, so heat will be lost by radiation through the skin. Secondly, the sweat glands begin to produce sweat, and as you sweat, the sweat will evaporate using heat that is in the body. And these two mechanisms would cool the body to the normal 37 degrees C. Now, if the opposite should happen, that is, the temperature drops, you go into a cold environment, and the temperature drops below the body temperature, specialized thermoreceptors which respond to cold will be stimulated, and these will send information via, of course, the sensory neurons to the controlling center, which is in the hypothalamus of the brain. This will be compared to the set point, which is 37 degrees C. Then the deviation will be calculated, and the output will be sent to the effector. The effectors will then generate the appropriate responses. And the response to cold will be the constriction of the vessels that supply the skin. So the blood flow to the skin decreases and very little heat is lost by radiation. Secondly, the skeletal muscles will begin involuntary contractions, which results in shivering. Normally, the involuntary contraction will generate heat that will warm the body. So the activities of the muscle and the decrease in blood flow to the skin would ensure that heat is conserved and the temperature is maintained at 37 degrees C. Now, another parameter that we can look at is the blood sugar level. Whenever we eat, food is broken down and the end products are absorbed. So sugar will be absorbed. And the plasma sugar has a set point. So if it goes above the set point, this is detected, and the beta cells of the pancreas will produce insulin. Insulin will make the cells permeable, and glucose will be taken out of the blood into the cells. And this would ensure that the blood sugar is maintained constant. If we take sodium, we have the renal system, which ensures that if we have excess sodium, it is eliminated. So that the homeostatic control component ensures that parameters or variables are maintained relatively constant in the body. The cardiovascular system ensures that our blood pressure remains constant. Renal system regulates the pH and the gases, concentration of gases. Integumentary system plays a role in temperature regulation. So all 11 systems ensure that the internal environment is maintained constant. Now, the homeostasis is maintained by two basic strategies. These are feedback and feed-forward mechanisms. Now, feedback control is a type of mechanism or control where the product of a reaction determines the cause of the reaction. A reaction begins and some products are generated, and these products will determine the cause of the reaction. Now, it could be a positive feedback reaction or control, or a negative feedback reaction. I want us to look at the negative feedback. Now, negative feedback occurs when a change in a controlled variable generate a response that opposes the change. Again, a change in a controlled variable generating a response that opposes the change. What we saw with the body temperature is a typical example of the negative feedback. We realized that as we got into the hot environment, this generated a change in the body. The body temperature became higher than the set point, which is 37 degrees. So the body generated a response which opposed the change. And what are the responses? Vasodilation of the blood vessels in the skin, which ensures that heat is lost by radiation, and the sweating and subsequent evaporation of the sweat that ensures that the body temperature is cooled. So the temperature remained the same. The changes generated a response which opposed the change. Right. Now, if we look at this hand of mine, right. Now, let's say it's supposed to stay this way. Right. It's supposed to stay in this position. It's supposed to stay in this position. Now, if there should be a factor that is moving it in this direction, right, and I ensure that it's brought back to its normal position, this will be said to be a negative feedback. There is a change, right? This is the change. And I'm forcing to bring it back to its original change. I'm opposing the change to maintain the status quo. Then I have a negative feedback response. Now, negative feedback ensures equilibrium, right? It ensures equilibrium. It ensures that the internal environment is maintained constant. The internal environment is maintained constant. Now, I'll give you another scenario. I don't know if your lecture room is air conditioned. If it's not, forget about it. But let's say your room is air conditioned. Now, this air conditioner has a thermostat. So you go to your room, you switch on the air conditioner and set it at, let's say, 20 degrees C. Right. At the onset, you hear the fan working. The air conditioner will be working, and the temperature in the room would be brought to 20 degrees. Now, when the temperature in the room is 20 degrees, the fan ceases working. Right? The fan will cease working. And it stays so till the temperature in the room begins going up. Then the fan will start again to bring the temperature back to 20 degrees. That's an example of a negative feedback response. Does it make sense? Does it make sense, please? Good. Now, about 80%, okay, what, Modupe, I'll go back again. You get an air conditioner with a thermostat. You switch it on and decide that you want the room temperature to be 20 degrees, right? So the fan will start working and the room will begin getting cooler and cooler till the temperature in the room gets to 20 degrees because you've decided that you are setting it at 20 degrees. Now when it gets to 20 degrees, the fan which regulates the condenser to bring out the cool air will cease working, right? Because it's controlled by the thermostat, which you've set at 20 degrees. So the room remains at 20 degrees, but because you are not generating cool air, you realize that the room temperature begins to climb. goes about 20 degrees. The moment it goes above 20 degrees, the thermostat will detect this and the fan will start working to cool the room to 20 degrees. Right. So a change has been detected and the change is being opposed to bring it back to the set point. Right. 20 degrees is your set point. So the room temperature is maintained at 20 degrees as long as that is your set point. I hope this explains it, Modupe. Modupe, feedback. Is it clear now? Good. Now, I'll use the remaining 5 minutes to go through, okay, this is the negative feedback response, right? Now, this is our variable, which is in homeostasis. It's in homeostasis, that's the set point. Now, if there is a stimulus that will cause an imbalance, this is what we have. This imbalance is dictated by the receptors, right? And the receptors will send the information via sensory neurons to the control center, right? So the receptors will send inputs to the control center. The control center would compare the input it's receiving with the set point and generate a deviation. So it detects that there is a deviation. It generates an output. So the output is sent to the effector. And the effector will bring about a response that will correct the change, right, to maintain homeostasis. So this is the negative feedback and how it controls homeostasis. Now the second mechanism under feedback is the positive feedback. And in positive feedback, a change in a control variable would generate a response that would amplify or reinforce the change. We saw that in negative feedback. In negative feedback, the change would generate a response that would counteract or oppose the change. In positive feedback, a change would generate a response that would amplify the change or reinforce the change. Now, going back to my example, my hand is supposed to be staying this way. Now, in negative feedback, we saw that when it's moved this way, I'll oppose the change and bring it back to its original position. Now, in positive feedback, if there is a change, instead of bringing it back to its original position, it rather moves away from the original position. So the change is reinforced, right? And normally, positive feedback brings about disequilibrium. Even though, yes, it brings about disequilibrium. And it doesn't play a direct role in homeostasis, but it's very relevant in some negative feedback processes, and it's important in some physiological processes, right? Positive feedback, a change in the control variable will reinforce the change. Now I'm going to give you examples of positive feedback in the human being. I guess you know about ovulation. That is where the ovum is released from the ovary during the menstrual cycle. Now, maturation of the and release of the ovary depends on the ovum depends on estrogen. Now, estrogen at the onset of the maturation of the follicle is in a low concentration. Now, as the concentration increases, it now will cause a negative feedback in the release of luteinizing hormone. Now, the luteinizing hormone produced from the anterior pituitary is responsible for the ovulation. Now, estrogen secretion as it continues will cause a negative feedback. So luteinizing hormone secretion will decline. Now, as the concentration of estrogen increases, instead of causing a negative feedback response, it causes rather a positive feedback response. It causes further increase of luteinizing hormone, which results in what we call the LH surge. And the LH surge is responsible for the ovulation. Without the LH surge, there is no ovulation. So increased estrogen causes increase in LH, and this will induce the surge, the ovulation. Now, another example of positive feedback is during parturition. In our next meeting, we'll continue with we'll go over the positive feedback.